This is a tutorial to the basic features of Mesquite, how you approach it if you're coming at it for the first time. We're going to start with two data files. I'll just open them both up to show you what they look like. This is a FASTA file with DNA data, and this is a file, a nexus file of beetle morphology. The first thing you want to do is to actually get your data into Mesquite to work with it. So we'll try opening one of those files, and we'll just start with the beetle morphology file. Nexus files are the default language of Mesquite. So if you just simply ask to save, it saves as a nexus file. This is how that file got opened. You'll see the taxa here, the characters across the top, and the characters with their character state names. There are two critical windows in Mesquite. One is the character matrix window, which could be molecular data, for instance, and then a tree window. And you'll notice over here on the left is what we call the project panel, which summarizes the resources of the file, including a matrix and a block of trees. And both of these are related to a particular set of taxa or block of taxa, 19 taxa. So if we want to look at the trees in this block, we can touch on this and it says view trees. So this is the tree window. So we now have a tree window and a character matrix window, and we can do a lot of things. We can try manipulating the tree. We can hit undo there. By the way, there are very few undos in Mesquite because it's super complicated to try to account for all the different things the user can do. Sorry for that. So we suggest you save your files frequently. And in this window, the tree window, we can do things like reconstruct ancestral states. So we can say we'll use parsimony. And this one is a fairly basic character, but we can scroll through the other characters and see what it looks like. I'm going to change the form of the tree here so that it's a little easier to see those. And here we go. This is balls and stick. In the tree window, compared to version 3, we've split up the menus a bit. We have an alter menu where you can make changes to the tree other than ones that you do with the cursors. We can change the form of the tree, and that includes things like the orientation. We can change the color of things. So it could be, if you just really love a goldenrod background, there you go. And you can change things to do with the text, the names and so forth on the tree. And I'm just going to change the text on name angle here. The tree window not only allows you to see the trees that are stored in the file, but you can ask to see other sources of trees other than the ones stored in the file, including simulated trees. So we could ask here, for instance, tree source, simulated trees, coalescent trees, or birth-death processes trees. And what would be shown here would be those sorts of trees. Let's get back to the matrix window for a second here. There are colors across here. If you don't like that, you can go to the display menu and you can see different ways to change colors. A color of character names background, let's just say no colors. If you want to do things like edit the state names, because this is morphological data, we can say edit state names and we can edit them here. So that is the matrix. But there are several other absolutely critical windows in Mesquite, and they're the ones that list the various pieces of the resources. So the taxa window, the list of taxa window, here has just the list of taxa, and you can look at various features of them. The column that's there by default is whatever group they're assigned to. You may very well have assignments. You might have the outgroups versus your study group. You may have subgroups of your study group that you consistently want to refer to. And we find it super useful to designate these groups and give them color because it helps you keep track of a lot of things. I'm going to do a little trick here to designate this set here as a group. I'm just going to go here. I'm going to select this here. And I'm going to say new taxon group for clade. And let's just give it this. And these are David's beetles, not my spiders. And I don't know what to call them, so I'll just call it the green clade. And so there we go. We've got the green clade now. And if we come back here, we can see it's these. So we can give them names. And among other things, if we're back here in the tree window, and let's suppose we close this off, we can do things like color branches by taxon groups. So now this is just one of the groups compared to the other ones. And when you do this, you can end up, as you're looking through various trees, more quickly spot what are the the known bits that you have. To get back to the taxa window here, in the list here, we can do things like change the names. We can go through some things to go through and sort of automatically search and replace various things in names as well. One of the columns that I tend to like to use is called alternative names, where we can replace the alternatives by the names, for instance, and now we can edit this and say, uh, uh, modify it in different ways and then later swap them back. And this can become a storehouse for a set of alternative names that might be, for instance, the names that you use in certain of the trees that you then later want to reread. So that's the taxon list window. The list of characters is another critical one, and you'll notice that these characters are already assigned to groups. These are the adult external characters, these are the male genitalic characters, and so forth. You'll notice here the parsimony model that's used is designated and so forth. So here you can assign various qualities to the characters. There's also a list of character matrices, and this isn't too interesting if you've only got one matrix, but if you've got 500 matrices, then you can start manipulating those there. You can also have a list of trees. So if you had 500 most parsimonious trees or 5,000 gene trees or whatever, you can likewise look at these and manipulate them. So these various list windows are rather important. You can get between these critical windows in lots of different ways. And so one of the ways is what I've been showing you, is to go here and say list and manage trees, view trees, show matrix, list and manage characters, list and manage taxa, and so forth. But there are also ways to get to them through menus. So for instance, you can get a new tree menu this way, a new tree window this way. And if you wanted to show trees that are already in the file, you say trees from source and then choose stored trees. You can also show the list of taxa here, a list of trees here, a list of tree blocks if there's more than one tree block, and so forth, and various other lists here. You can show the character matrices through a matrix, through a menu as well. So you can say character matrix editor, choose which one you want. You can choose lists of characters, lists of character matrices, and so forth. Also, if you're, say, here in the character matrix editor, you can get between things using some of these little buttons. So for instance, you can say C to go to the list of characters, and back here to go back to the matrix. Let's try the other file, the FASTA file. Now here, as I said, Nexus is Mesquite's native language. And so here it's found a file that it's trying to figure out what this is. And Mesquite's not quite smart enough to figure out exactly what the file format is. We let you tell us. So basically here it says it's a FASTA file. So let's choose this. And now it wants us to immediately save it. I'm just going to save it as a .nex file. And now we have these data. You can see that they're not aligned. Luckily, Mesquite has various alignment programs built in, such as MAFT and Muscle. So we'll just do a quick MAFT alignment. Ooh, that looks rather messy. In fact, the reason it's so messy, this is UCE data, and these are the flanks that are in rather rough shape. So I'm just going to do a quick little thing here to trim the sequences by trimal, and we'll just do that. And now we can hit the bird's eye view to get a more compressed view, and here's what it ends up looking like now. Not only can we import data of a different format, we can also export data to other formats other than to Nexus. And so these are some of the things we can export for, and some of them are very tuned to particular purposes. We can export Nexus for different programs like Beast and Mr. Bayes, and that includes the ability to export fused matrices, such as here, that include, say, molecular and morphological data. With DNA data, there's a lot of other things to think about, including things like codon positions, genetic codes, if you want to look at it with amino acids, and so forth. So depending upon the data type, you end up having different available options. In these windows, I often use these tools, the magic wand and the sort. So for instance, if we now say, I want to see the sequence length of these different things, I can ask to look at, I often color cells because I want to see who's weak or who's strong in these things. And so I might, for instance, choose anything that's there or below a certain number and see how many we have that are as bad as that or worse. This tool can sort, so we can basically say like this and sort them this way. We do a lot of painting just to keep track of things. And so here we can, for instance, say we're going to assign some of these taxa to be blue for some reason. It's all up to us why we do it. And then if we were to then show a quick tree, which we will just ask RaxML to do here. RaxML here is one version is built in. We can get us the tree. If we ask, we can see, for instance, color of taxon names, color by assigned color, which these are the blue ones that we had shown. One can then take the tree image and save it as a PDF. And so here I'm just going to save it as tree ACA. Then that tree image can be edited. We'd want to go in and crop this out. We could change font sizes, although we could have also done that in Mesquite. Usually you can go in with a vector graphics program and make all the adjustments that you need. There are a lot of different tree forms available. Of course, there's things like the circular tree and good old diagonal, which doesn't look very good when you have branch lengths like this. We can also do things like this. There is a lot more to show you, but this is just the barest introduction to get you used to things. And one of the things that you can struggle with is to figure out what is possible and what things do. And so we try to give you feedback. As I pass the cursor over these tools, you'll see down at the bottom, it tells you what the tools do. And so this area here is sort of a critical one for getting feedback. Another thing that you can do, some of the menu items, such as here, this is the include and merge. What these do, include and merge, is that if you've already got data in a file, but you want to add more data to it from another file, if you want to add trees to it and so forth, you can either use include file, which reads it in the other file as if it had been part of the first file that you already had, or you can use link file, which does the same except it leaves it as a separate file. Or you can use some of these other ones to deal with more difficult cases. But it turns out we also give a little explanation here as to what these things are. So if you get confused as to what these things do, there's a few menus that have these explanations built in. The other thing you can do, and I do this myself because I forget where certain things are, is if you go to View, Menus and Control Explanations, and Mesquite will compose a web page that tells you about all the available menus that are currently there right on the screen, as well as gives you a little summary of what these things are, and then you can do searches in there to see what might be relevant to what you're looking for. That's that.